Going to talk about genes. Actually, I'm not an expert of genes, but as long as the basic definition of the genes is concerned, I would like to go with that because then in future we may try to connect this idea with the framework of quantum mechanics and try to make some correlations with the very concept of genes that we have. So what we'll be looking for is we'll try to understand what genes are basically in literal sense. And here, obviously, we are not going to talk about any kind of mathematical equation. We are not going to talk about any quantum mechanics at this preliminary stage. We are just first trying to understand the very basic concept of genes that's purely coined in the field of biological sciences. So, so how to start with is the right idea would be that we would like to have a clarity on these basic concepts of genes. I believe that we would like to understand in literal sense what genes are, and once we get the concept of the genes, then we will try to see how we can allocate some of the concepts of physics in context of the genes, like probability, uncertainty principle, superposition, and certainly these things here we are trying to link these basic formalism of quantum mechanics in describing the nature of genes. So, guys, here we are, and here we will be talking about the genes first. But the core idea is that we want to talk about the quantum mechanics of genes, and that's what will make this understanding better for us, because we are trying to understand it in the framework of we are trying to understand it in the core framework of quantum mechanical laws. So let's start with this. Oh no, I think we have lost the light. And so what we have is we will be talking about we'll be talking about the genes. So the very first question arises. The question that arises here is that what is a gene? And a very plain answer to this question can be that a gene is basically a part of your body, a part of your body that carries information. This is how we can describe genes in the simplistic forms. Now, what you are doing here, you are not you're not coding down the information like a thought. No, there's not a thought process going here, and obviously it's not the information like a memory that is executed in the brain. No, the gene that we are describing here, what it represents, as I said. So what we have here, we have the biological information. It is the biological information that's coded in these genes. So biological information, it's an information that helps your body grow, that helps your body repair, that helps your body function, and that helps your body respond to its surroundings. So this is what we can argue in the simplest way what genes are actually doing. So if we freeze it to the humans, every human cell, maybe except a few special ones, contain these genes. And what these genes do is these genes help decide. They help decide how cells are built, how the framing of the cells is done, and which substance they can produce. So see the dictation of this very fundamental part of the organ, and when these cells should act, and when they should remain silent. So the trigger is coming from the genes in a very, very canonical sense. So what I can say here is that I can translate down this thing in the simplest language. If I translate it down into a simplest language, a gene is a biological set of instructions. It's a biological instructions basically that helps a living cell know what to do and when to do. This is what we understand. This is what we understand about the genes. Okay. Now, to proceed from here, this is not the only job of the genes. What we have is we have a point where genes are located, and we have a point genes are not freely floating like electrons. They're not floating like atoms and delocalized electrons. And they are not spread randomly. They are written. They are written inside a very long molecule, the long molecule that we know as the DNA molecule, deoxyribose nucleic acid. So DNA exists inside the nucleus of almost every human cell. And to be precise, if we stretch the DNA from a single cell, it would be nearly 2 meters long. Its length will be 2 meters. And yet it fits inside the space far, far smaller than a grain of rice, than a grain of dust, than a grain of sand. This alone tells us something important. This alone reveals us something extraordinary. Genes exist in a world where smallness, packing, and precision matters. And these things matter enormously. These things matter enormously, tremendously, extraordinarily. Now, if we again try to understand the basics of the DNA, we want to learn it. And what DNA is? DNA is basically, I have lost the light, it's basically made of four basic chemical units. There are four, one, two, three, and four basic chemical units. And you can think about these chemical units as letters: A, B, C, D. Or you can think about them in terms of any kind of symbolic presentation, X, Y, Z, whatever you can have. So here we have special letters A, B, C, and G. So these letters, now what does these letters reflect? These letters, A, T, G, C, these letters are arranged in a very, very long sequence. It's just like the words. You have a word is formed out of letters, okay? If we think in terms of a word is formed from letters. And for genes are formed from these symbols. They are formed from from all these four symbols that I talked about. But unlike human language, DNA is physical in nature. You can actually touch it. You can actually touch it. You can touch it directly or indirectly. You can Okay, so I'm sorry. So we are talking about the combination of these. We're talking about the combination of these letters, and it's the combination that matters. It matters. It gives them the meaning, and it gives them the nature, the way they act. And the question here arises now is that what makes a gene different from DNA? Because we have DNA, how we can differentiate between a gene and a DNA molecule? This is very crucial. We want to understand it. We want to understand it at the basic level. As I said before, I'm not an expert of biological science, but as a piece of information, I'm sharing this thing with you. Because I want to make a connection. I want to make some connection with quantum mechanics. A gene is a readable section. A section that can be read. And not every part of DNA, we can say, is a gene. Only those regions that can be read, that can be used by the cell, are called genes. This may be the simplest way to realize what genes are. So to interpret it, what am I trying to tell here is that, so a gene is not defined only by its position. No. Basically, it's defined by its function. The function that the gene does. Now, if we think about a segment of a DNA, we have a DNA molecule. If a DNA segment is never read, is never used, is never activated, then practically it is silent. It's not functioning. It's the silent part. So what we can say here is that a gene exists, or I should say, a gene exists only when it is functionally recognized by the cell. This is what makes the part of that DNA molecule as a gene. So this is the idea. This idea will become very important in our future understanding, where we will try to make a connection. We will try to build build a connection to the mathematical, quantum mechanical understanding of the systems. Now the question arises: how does a cell read? So the cell has to read. What cell is reading is genes. It's not reading the whole of the DNA molecules. Cells do not have eyes. They do not have consciousness. So what is it that makes them masters in recognizing the genes? What they do here is they use molecules. They use basically special molecules. What am I trying to tell is that special molecules move along DNA. They move along this molecule deoxyribose nucleic acid. And these molecules, they are temporarily attached to it. What they do is they sense the patterns. They copy the information. And this process of copying the information is called gene expression. It's like a mathematical equation is being read by these special molecules roaming with the DNA molecules. So gene expression happens. It happens in steps. Step 1, step 2, and so on and so forth. The gene is identified and the gene is copied. It is copied into a temporary message. See the beauty of that, beauty of the things that are happening there. And imagine the depth. So what is happening? The message, this message is used to build your complex proteins. And proteins are built. Proteins then do the real work of the cell. They do the actual work of cell. But the point is, it's coming from copying, and what is copied is only some important parts of the DNA, what we call as genes. So genes do not act directly. They are not directly taking the part, but they are the prototypes. They act through the chains of physical events that are happening as long as there is biological life. As long as there is biological life. Genes do not work all the time. And they cannot do that. This is one of the biggest misunderstandings that we have. A gene, we can say a gene is not always active. What am I trying to say is that there are many genes which remain silent, and they remain silent over years. Some genes work only during early developments. And you have the genes that remain silent in that stage. And some genes activate only under panic, under stress. See the beauty of flexibility that's going on there. It's purely quantum mechanical. So genes are conditional. They respond when they are needed. They act when they are needed. And so what they are doing there when they are not needed, they wait. They depend on signals that are coming from within, that are coming from outside this biological life. And this is what makes genes very different from rigid machines. There is that probability of actions going on, but these probabilities are well defined. They behave more like options than commands. They behave more like the collapse of the wave functions. So genes, they can be influenced by the environment, and these genes are sensitive. And at the same point of time, I'll tell you that genes do not work in isolation. They cannot work in isolation. We have that basic quantum mechanical framework for that. Temperature affects them. The chemicals that are surrounding them, these chemicals affect them. The nutrition, the food that we eat affects them. It makes an impact to these genes. The radiation that's coming from the outer world, the radiation affects them. It makes impact on them. So genes they're very much sensitive. So genes are sensitive physical systems. What they do is they sit at a scale where tiny changes can matter. A small minor change can matter. And this sensitivity is, what I can say is, this sensitivity is exactly where the classical certainty begins to fail. The classical Newtonian certainty that gives us mobile phones, that gives us computers, that gives us the moving satellites, they fail there. These genes, they act as information carriers. Their responses are dependent on the tiny changes. They don't respond classically. Genes can store information, but this information is not the energy. We're not talking about the energy that they can store. It's not the mass. They're not storing the mass. They're not adding the atoms and molecules to them. And it's not the force, even the force that you can think of that you're adding to the genes. So what information means here is that information is about the arrangement that we are talking about. So the information that you can store on these genes, these informations are stored in terms of the order. They are stored in terms of the pattern. They are stored in terms of the constraints that are being executed. So we are talking about genes. I wanted to actually put it on, but I have lost the light. So a gene is therefore a physical pattern. And this physical pattern should remain stable. But the stability of just this physical pattern is not rigid. It's not like a classical rigidity. Too much rigidity and it cannot adapt. It's impossible for the genes to adapt any kind of information if it is rigid. And at the same time, it has not to be flexible. Too much of flexibility and it loses the meaning. It doesn't make genes. So what's happening? Genes survive. They survive in a narrow, I should say, a window. They survive in a narrow window between these two extremes, rigidity and flexibility. We are basically trying to understand what genes are, how they work, and what is their nomenclature, and why genes are never perfectly predictable. At the level of genes, at the level of these four letters that I talked about, at this level, molecules, they oscillate. Molecules vibrate. The bonds, they fluctuate. And what's happening there is you have interactions. And these interactions are probabilistic. These interactions are like a collapse of a wave function. And nothing is happening at that level with absolute certainty. At that level of these four letters, nothing is happening with certainty. But at the same time, an overall order emerges out of it. An order is emerging out of it. And this tells us something strange. This tells us something profound. This tells us something realistic. What's that? Genes operate successfully in an uncertain physical world. This can be the right argument for today's talk. They do not eliminate the uncertainty. Their stability is a result of that uncertainty. What leads us to that Heisenberg's uncertainty principle? They manage it. They do it. They obtain those stable states. They obtain those the stability. They obtain their stability from probability. They obtain their stability from uncertainty. They obtain their stability from the superposition. And what we have done, what we have understood till now, we have understood something essential. I think we have understood genes without any mathematics in it. It was like that I was quoting the history of these genes, the historical events. And to be precise here, you certainly need advanced biology. You certainly need mathematics. You need physics. You need laws of physics. No doubt, we now know the genes are basically the sections of these DNA molecules. Okay? And they are the carriers of biological information. The biological information in terms of these letters. They are conditional in action. All of the genes do not act simultaneously. Some of the genes remain silent, some of the genes go on working. They are sensitive to the environment. They are sensitive to the environment surrounding them. They are physical, but at the same point of time, they are meaningful. So from here, in our next interaction, we will try to go deeper. Here we try to understand the basics of the genes, what the genes are. In our next interaction, we will ask about why probability matters. We will try to go deep into the concept of genes, and we'll try to link them to the very fundamental laws of nature, the timings, the uncertainty, the probabilistic nature of the universe, how it is impossible to ignore the very negative principle of the quantum mechanics known as Heisenberg's uncertainty principle, because that becomes a basis for the stability of your molecules. And this is what we'll be looking for. We'll see that the behavior of the genes, what they do is they naturally invite the language of quantum mechanics. They invite the laws of quantum mechanics